Hi, I'm Suzanne. I'm an application scientist at Nexcelom Bioscience, and today I am doing a video demonstration of how our K2 and X2 cellometers can help you count really accurately and look at viability of all your different cell types. So I have an X2 here, which is one of our instruments. It's attached to a laptop. If you are getting one of these for evaluation, you will get this in a box along with a laptop with the software that's preloaded onto the laptop. You'll have a plug that connects the cellometer to the electricity, as well as a plug for the laptop, and also a USB cable that connects the cellometer to the laptop. They're the only wires that you should get with your instrument. Connect all of those up, and then there is a switch on the back of the X2 which will turn it on. The light will come on in the front like this, and then you're ready to get started. So I've loaded up the software already, which is what you can see on your screen there, and I'm going to show you basically how it works and how you can use it in your lab. It will come with these plastic disposable slides that look like this. They have a film on the front that you need to peel off like this, and a film on the back that you need to do the same. So peel those off before you get started. Try not to touch any of the clear counting chamber area. You can touch anything that's around the edge on that white area there. So I'm just going to place this down on a Kim wipe or tissue and show you how those work. So once you have the X2 switched on, you can then load up your software, which should have the little Nexcelom icon there, and your software should load up and look something like this. So I'm going to talk a little bit more about the slides. You have two counting chambers, one here and one here in each of your slides. You can see that there's a little hole. If I hold that closer, there's a hole at the top and the bottom of each counting chamber, and what you can do is you can pipette your cells with trypan blue or with a fluorescent dye into the hole. Once you pipette in, it should fill the chamber and look something like this. So our X2 and K2 cellometers are both fitted with a bright field channel and a green and a red fluorescent channel as well. This means that you can look at viability using trypan blue in your bright field only channel, or you can look at viability using the green and red fluorescent channel. We suggest you do this especially for really tricky samples, and we have included some fluorescent viability stain. It will come in either a bottle that looks like this or like this, and that should come included in your pack, whether it's an evaluation pack or an install pack. How this works, in short, is your live cells will be stained green and your dead cells will be stained red. Any bits of debris, anything without a nucleus or without DNA, is not going to be stained with anything at all. So there's no more questioning, is this a cell, isn't this a cell? It really does help you distinguish that. So how it works is I've just pipetted 20 microliters into a tube. I'm also going to pipette 20 microliters of my cells into the same tube. You can use an Eppendorf tube or a Falcon tube, whatever's easiest. Give it a really good mix, but you don't actually need to leave it to incubate at all. All you want to do is take 20 microliters of that mixture and take one of your pre-peeled slides that we showed you before, push your pipette into the hole and fill that chamber with your cells. So once you have that, make sure you do use 20 microliters. Don't try and put any more in, it's just going to spill over the edge. You can place that into the instrument and you should be able to see your cells come up on the screen like this. So these might need a couple of minutes just to settle down a little bit. If you don't see your cells immediately on the software, what you can do is, I will just stop this preview, press the preview B1 channel, and they should come up for you. What our X2 and K2 cellometers have is they have a focus dial on the side of them here. This allows you to go through the different focal planes of your cell sample, like so. You can go right out of focus there and back into focus. What we want to see is a nice dark edge to your cells and a nice bright center. You don't want to focus like this, that's too far out of focus. You also don't want to choose the focus type that makes them dark in the center like this. So try and remember a dark, crisp edge and a nice bright center. So once you have your cells all focused with a nice crisp edge and a bright center, what you can then do is you can look and choose what assay you would like to run. So I am using just some ordinary baking yeast, just as an example. So I'm going to use my yeast assay with AOPI, that's the fluorescent dye. If you were using PBMCs or a mammalian cell line, a cancer cell line, I suggest starting with this PBMC preloaded assay for you with the AOPI, so with the fluorescence. But for now, I'm going to start with using the yeast experiment. And after I've focused, which I already have done, I'm now going to look in my fluorescent channels. So what I'm looking for is some nice green live cells, which is what I can see here. I don't want the focus to be overexposed, but I want to be able to see my live cells. I feel like we can see them quite nicely here. So I'm going to stop the preview, and I'm also going to preview the red channel as well. So the red channel takes a little bit longer, the exposure is a little bit higher, so hopefully we can see some dead cells as well, which we can do here. Again, the background's low, the exposure is not overexposed, but we can certainly see that there are some dead cells. If you wanted to change the exposure, there are ways you can do that here. You can move this this pointer down or up, depending on whether you want to change the exposure to make it lower or higher. But if you are happy with it, then you can press the count button. So what the instrument is going to do now is it's going to take 4 different images. It's going to take an image in bright field, and then in green, and then in red, of each of your 4 different areas. It's almost like looking at the 4 corners of a hemocytometer. So it will take those, it should take less than a minute to do the complete count. You will hear it changing from between the green and the red channel each time. And those images are named A, B, C and D. And you can flick between the images using this little function after it's been taken. So once all the images have been taken, what the software will do is it will add up all the green cells, the live cells, and all the red cells, which are the dead cells, and a results page will pop up right at the end. It will tell you the total number of cells it's counted, the total number that are live, and the total number that are dead, as well as your viability of your sample. It will also give you concentrations of those as well as diameters. My yeast are around 4 microns in diameter, so if that's something you're interested in, you can monitor those as well. So if you wanted to export your results and your images, there is an export button here. You can save your images as they are. You can save the counted images. You can save the combined images, depending on which ones of these you check. You can also export your data as a Excel file if that would be more useful for you. Just press continue there and that will all be exported for you. If you don't want to export it, you can close down your results page, and then what we can do is, these are your images that you can flick between. So we have A, B, C and D. Those are the images in Brightfield, and then what we can do is we can look at the images in green to show your live cells, or red to show your dead cells, or what might be more useful is if you click the combined image there, and then you can see all of your live and all of your dead cells in one image. What you can also do with this little 1, 2, 3, 4 button is zoom in on a quadrant of your image. So let's just zoom in on something up here, and then let's also look at the counted image as well. So what I'm now looking for is for all of my live cells to have a little green circle around them, and all of my dead cells to have a little red circle around them. Let's check out another image. Let's go to A and zoom in a little bit as well. What I also want to see is I want to see that any that are in clusters are being segmented nicely and being declustered appropriately. And if you are happy with the settings, then I would save your results. To get back to that results page, there is a button up here. That is the very basics of how the Cellometer works. I want to show you a couple of other functions that you can look at as well. So if you close the results, we've got two other buttons up here. This first one on the left I've shown you will get you back to that results page. The second one is a calculator. What you can use that calculator for is, after you've determined your live cell concentration, you put how much volume you have in there. So I'll pretend I have 10 mils of volume, and my target concentration is 1 times 10 to the 6. You can change that. Say I want a target concentration of 2 times 10 to the 6 in my newly seeded flask. If you apply the change to that, it will tell you how much diluent you need to add to your original 10 ml volume to get your desired concentration. If you don't want a concentration, but instead you want a target number of cells, say you're seeding a 96 well plate for instance, and you want 10,000 cells per well, you can add that, apply the change, and it will tell you how many microliters you need to add to each of your wells to get your 10,000 cells per well from your original sample. So that's the calculator. The next one is a size histogram, which can be really useful if you're monitoring cell size. Your your live cells will be the green bars, and your dead cells will be the red bars, and it just shows you the size distribution. If you wanted to add a gate to that, for instance, if you think, well, I'm looking at yeast here and I think anything above 10 microns is definitely not a yeast cell, so I'm going to put a gate on that. So I'm going to keep my minimum cell size the same. I'm going to add a 10 micron gate to the max box, add a new gate, and you will get your different results up here depending on what gate you put on. You can also clear all gates and get it back to the beginning. You can save, copy, customize this histogram as you wish, or print it. So the final thing I want to show you is how to create a new assay type for some of the specific cells that you're counting. I would start with this PBMC assay. It's really good for, I'd say, normal type cells. It's really a good starting point for that. If you want to create an assay for a new type of cells, you will click on this little edit button here. Okay, so at the top, click save as a new assay type. And let's say we're looking at CHO cells, so I'm going to save this as CHO with AOPI, so this doesn't overwrite any PBMC assay. Okay, the next thing I'm going to do is move down. The two boxes that you should be really interested in is this box and this box here. What we have here is, in the cell type drop down menu, we have a very long library of lots of different cell types that have been tried and tested on these cellometers, and what you can do is your cell type will probably be in there. You can click on it and use the determined cell type parameters, which is fantastic. I would certainly try that with both of your channels to start with. Select those, press save. So now you should have a new assay in your drop down menu, a CHO assay, which is appearing here, and you can see that the cell type, we've set it to CHO cells, and you can count your cells using those settings. What I would also suggest that you do is, if those settings are the default settings that we've tested CHO cells on before, but every lab's cells are slightly different. Don't be scared to look into it and change some of the settings. So if you feel like your CHO cells aren't being counted correctly, there isn't a nice red circle and a nice green circle around all of your cells after you've counted, you can edit the cell type. So here again, what I would do is I would save it as something new. So you can save it with your name maybe, to show that you've edited it and you're not overwriting anything else. So now it depends why the cells aren't being drawn around and identified correctly. It could be due to the size parameters, it could be due to the roundness parameters, or something similar. So let's pretend that there are some big cells in there that aren't being detected. So I think I need to change this size parameter from 38 to 50, which I can do there. If you think it's a fluorescent parameter, say your cells aren't bright enough to be detected, it's this threshold that you need to be looking at. The threshold is set somewhere between 0 and 100, and that is as a percentage above background. So it's currently set to 5% above background, so any fluorescent object that's fluorescing 5% above the background level will be detected as a positive object. If you think some of your weaker stained cells aren't being detected, we could lower that down to 2 or lower that down to 1, just to make sure everything's being picked up. Beware about lowering it too much. You don't want to pick up objects that really are background and not real cells, so just be careful with that. The other option I wanted to show you is this advanced bright field fluorescent mode. This is the one we get asked about the most, and I think this is the one that's really important. It's mainly for clumpy cells, or cells that are forming clusters of 3 and above. If you check that on, what that will do is it will use the bright field image in combination with the fluorescent image in order to really help decluster those clumps. So I would suggest take a look at this and switch it on if you're getting even small clumps. Once you've done that, you can save it, and you can see that my newly saved cell type is now appearing in this box under this assay type. Okay, finally, there's a couple of extra things on this side here. One is that you can set your sample ID, so you can call it whatever you want. You can name it with the date, anything like that. If you don't have to set your sample ID. If you do, it means that when you come to save your data, it will be given that sample ID. It makes it much more easy to locate. It will automatically be date and time stamped, so it just gives you an extra bit of identification. If you do a pre-dilution, so you're not just diluting it 1-to-1 with your fluorescent dye or your trypan blue, if you're doing a pre-dilution as well, you might want to change your dilution factor. So say you did a pre-1 in 5 dilution, that would give you a total dilution factor of 1 in 10. If you type that in before you do the count, that will automatically be taken into account when you get your results page. So just make sure you've got the dilution factor set to exactly what you need it to. And I think that is everything. In fact, the very last thing I want to show you is if you count something, then you change your assay settings or anything like that. You don't have to do a full retaking all the pictures of the same sample that you've already done. what you can do is you can press this little recount button here. What that is going to do, it's going to take the same images as you've taken before, and it's just going to apply your other settings to them. So all I did change here was that I changed the dilution factor to a 1 in 10, and that's changed my concentration on the results page. Everything else is the same. But yeah, you don't need to retake all your images once you've taken them once. And I think that's everything. If you've got any questions, the best email to get us on is support@nexcelom.co.uk. That email will go to myself. It will go to also our support team, and we can get any questions answered for you as quickly as possible. But in the meantime, I hope you really enjoy your instrument. Do get in contact with us if you need anything, and good luck with it.